welcome to Chapter 3 on genetics and topic 3.1, all about genes. All right, so here's a definition that you definitely need to commit to memory, and that is of a gene. So a gene has to be something heritable, meaning passed down from parent to offspring. So it can't be something that you acquire during your lifetime, like a tattoo or a nice haircut. Okay, it has to be something in the DNA, and it's a length of DNA, so not a whole section or not a whole strand, but just a section. And it influences a particular characteristic like eye color or blood type. Okay, so let's take a look at this cell here. Okay, this cell I'm guessing is in really early prophase because we see chromosomes, but I still see a nucleus here. Anywho, okay, this is a chromosome. And you already know that a chromosome consists of two sister chromatids. Okay, so sister chromatids, meaning this is one copy, this is another copy. These are identical copies. So this is obviously taking place after DNA replication. And chromosomes consist of segments of DNA. And on strands of DNA, we're going to find different lengths that code for different characteristics, and each one of those lengths is called a gene. Okay, so here we have some information on different species and the number of genes in their genome. So I want you to take a look at this data and evaluate this statement. The more genes an organism has, the more advanced it is. So take a moment to kind of think about that and evaluate that statement. Now, humans have over 20,000 genes, but only 23 pairs of chromosomes. So what that means is that on each chromosome, we're going to have several genes, okay? And each gene occupies what we call a particular locus, or loci would be plural. And a locus is just a particular position on a chromosome. So whether it's up here, or down here, or in the middle, whether it's on the first chromosome or whether it's on the 21st chromosome, okay? These all go into what we call a particular locus. Now we know that we get two chromosomes, right? These homologous pairs, one from the mom and one from the dad. And we know that we inherit a copy of a particular gene for each trait. Like let's say this is the gene for blood type. Well, that gene for blood type is going to be on the same chromosome in the same locus for the dad as it is for the mom. So genes from each parent for the same trait are found on the same numbered chromosome in the same locus. Okay, so let's take a look at this diagram. So here we have a homologous pair of chromosomes. So what does that mean? Well, that means one from the mom, one from the dad. Here is another homologous pair. Okay, so here's one, here's another. Okay, I can tell that they're not the same chromosome because they're not the same length, first of all, and there are different genes found on them. Here is a particular locus, okay, just a particular location of a gene on a chromosome. Now, we know that we inherit two versions of a gene because we are the offspring of sexual reproducing parents, right? So we inherit a gene from the dad and a gene from the mom. And sometimes they're the same, but they're not always the same message. And these versions of a gene are what we call alleles. So alleles are a specific form of a gene, like the allele for being able to taste bitterness versus the allele for not being able to taste bitterness, or if you're a flower, the allele for purple flowers or for white flowers. Notice that these alleles are just different versions of the same gene, like flower color, and they are, of course, found in the same spot or locus on the same chromosome. So while these alleles might be found in the same locus on the same chromosome, they may have a different message, and that's because it's possible that their sequence of base pairs could be very different. So let's say this is the allele for blood type on the mom's chromosome, and here is the allele for blood type on the dad's chromosome, they would be found on the same locus, but they might have a much different sequence of base pairs, and that's how they differ in their genetic expression. So a great example of how alleles can be different is the allele for mucus production in the lungs. And at some point in our human evolutionary history, some kind of a mutation occurred in that segment of DNA that normally codes for normal mucus production. So I had some kind of a mutation here. The new version of that gene, called an allele, is recessive. And so if a person inherits 2 copies of that recessive allele, then they will get this disease called cystic fibrosis, a mucus buildup in the lungs. Okay, so before we start here, I should point out that in your notes, I made a little bit of an error. There should not be an extra letter here, and this should be a G, not a C. Okay, but now that I pointed out how careless I've been, let's go ahead and take a deeper look at how these base sequences can really make a difference by transcribing and translating this. So you may remember that in transcription, okay, we are in the nucleus, and we're going to take a segment of DNA and use it as a template to make a segment of mRNA. So when we want to express a gene, okay, RNA polymerase, that enzyme, is going to come along and unzip DNA and use it as a template to make a strand of mRNA. So now, whenever it sees a C on the DNA, it's going to lay down a G on the strand of mRNA. And A would normally be complementary to T, but mRNA has no T's, it has U's. And so we're just going to go along and write the complementary base pairs as if we were that enzyme making a strand of mRNA. Now we need to go along and translate that. And so translation, of course, occurs on a ribosome. And the ribosome is going to read that strand of mRNA 3 letters at a time, called codons. So I'm going to go ahead and separate that into 3 letters at a time, so those codons. And it's going to signal for the tRNAs to bring in the proper amino acid sequence. So to do that, I'm going to use my table, G, U, G. So G, U, and then G down here is going to make valine. For this next codon, G, A, C. So G, A, cross-reference, then C, we're looking at asparagine. UCA is going to be up here. UCA is serine. And then AGG is going to give me arginine. And then ACU would be threonine. And then the last one, CCU, gives me proline. Okay, so for this one, you should be able to transcribe and then translate this on your own the same way that we did the last one. Keep in mind, however, that the same mistakes I made on the first slide are also here, so be sure to fix those. Okay, so as you can see from that last slide, changing just one base in that long sequence can really make a big difference in the amino acid sequence. Doesn't always, but it can. So changing a single base can change the amino acid sequence. Now remember that proteins aren't proteins until they're folded into their final shape. And all of these shapes depend on that amino acid sequence, because to go from primary into secondary, whether it's an alpha helix or a beta pleated sheet, all depends on the hydrogen bonding between those amino acids. And because different amino acids have different properties, they're going to fold into different shapes based on their interactions with each other. So if I substitute one of these in for a different one, okay, that has different properties, I'm going to get a different shape. And so instead of folding into the original shape, it can now be folded into a different shape and then alter the overall functionality of that protein. Now, like I said, it doesn't always work that way, right? So sometimes we can experience a change in the DNA that doesn't necessarily result in a change in the amino acid sequence. And that's because, as you saw from the chart, some codons code for the same amino acid, even though they have slightly different base sequences in those codons. All right, so let's put a word on what we've just been talking about. So we keep talking about base sequences changing. Well, anytime that you have a change in a base sequence, that is what we call a mutation. It can be just one letter, it can be multiple letters, okay? And mutations are very rare because DNA polymerase and RNA polymerase are the bomb. They're pretty good at making sure that whatever base they lay down in the new strand of DNA during replication, that those bases strictly adhere to that complementary base pairing rule. So that's kind of one of the great parts about DNA. Its structure ensures the proper copying because A can only bind with T. So that complementary base pairing usually prevents any kind of mistakes from happening. Now, every now and then you can get a mistake where the wrong letter is inserted. It may or may not change the amino acid sequence as we've seen, but again, these are called mutations, pretty rare. Now, mutation kind of gets a bad reputation, right? So some mutations are bad, like we just talked about cystic fibrosis, but not all mutations are bad. Some mutations are really good. It can actually give the organism a better chance of survival. And so if that's the case, then that mutation may make that individual more likely to reproduce. And if it's more likely to reproduce, it's more likely that that mutated gene is going to be passed down to its offspring in future generations. So we know that there are some individuals who have a mutation in one of the genes in their immune cells, and that mutation causes the receptor protein on the outside of that immune cell to become deformed. And since that receptor protein is deformed, the HIV virus can't bind to that cell. And so because of that mutation, this person is immune. It's not able to get the HIV infection because the virus literally doesn't fit onto those cells. So here's an example of a good or beneficial mutation. And so just kind of something to keep in mind that not all mutations are bad. Now, how can we decide when a mutation is going to be passed along and become more common? Well, if it gives the individual an advantage in their environment, like better able to get food, more likely to gain space, and it makes it more likely for that individual to reproduce, then the mutation will become more frequent. And so this part about reproduction is really important because mutations are heritable. They're in genes. So let's say that you're a bacteria and you have a mutation that allows you to be resistant to antibiotics. That's going to make you more likely to survive and therefore more likely to reproduce. Or if you have some kind of a mutation that makes you like super sexy to all of the opposite sex, then you're more likely to reproduce and pass along those genes. So that's kind of one of the ways that we can tell if they're going to become more prevalent. Now, when these mutations are successfully passed from one generation to the next, that's how we get a new allele. So let's say there's a mutation that turns ordinarily long ears into cute new short ears, and those short ears give an individual a greater chance of surviving and then reproducing. When that allele gets passed down to future generations, or when that mutation rather gets passed down to future generations, that's how we get new alleles. So for a good portion of the human population, okay, lactose intolerance is completely normal in adulthood. So in your DNA, you have a gene that produces lactase. And lactase is an enzyme that breaks down the disaccharide lactose into its monosaccharide components of glucose and galactose. And glucose and galactose are small enough to be absorbed into the bloodstream through the walls of the small intestine. So if we don't have this lactase, we can't break down this lactose. And so if you drink something like milk with lactose in it and you can't break it down, that disaccharide then can't get absorbed by the small intestine, and it makes its way into your further down into your intestinal tract, and it gets broken down by bacteria and causes all kinds of discomfort, acid, gas, okay, cramping, and this is what we call lactose intolerance. So normally in adults, the gene that produces lactase turns off, and that's because in our evolutionary history, humans were only breastfeeding and therefore consuming lactose when they were small children and babies. By the time they made it to adulthood, they weren't consuming anything with lactose in it anymore, and so that gene turned off. At some point, about 10,000 years ago, a mutation happened, allowing that gene to remain turned on and to continue to produce this lactase throughout an organism's entire life. And that gave them, those individuals that had that genetic mutation, a distinct advantage. Because now instead of having to be nomadic and traveling around with herds or gathering vegetables, they could keep something like a dairy cow or another animal that produces milk and then have a sustainable food source all year long. And so then that became, in those areas, a very common mutation because it gave them an advantage. One of the examples of mutations and their expression that you need to know is that of sickle cell disease. So sickle cell disease is involved with hemoglobin, and you may remember that hemoglobin is a quaternary protein made up of 4 polypeptides. So each of these polypeptides has its own gene, so its own segment of DNA that codes for that particular polypeptide sequence that makes up the protein. Well, there's a mutation in one of those genes that causes one of the polypeptides to be misformed. And the way that that works is through a substitution mutation. And you do need to know these particular amino acids. So in this amino acid sequence, so what's supposed to go in here, there's a whole bunch of things, but I'm supposed to have here glutamic acid. It's supposed to be in my original DNA sequence, okay, GAG, and then its complementary CTC strand. Well, at some point, there is a substitution mutation, and that causes the amino acid valine to be inserted into the amino acid chain. And we know that when we change the amino acid sequence, it changes the way that that protein folds into shape. And here we get this phenotypic result of these sickle-shaped red blood cells, and that has to do with the way that the hemoglobin has formed. Now, this gene is a recessive allele. So in order for a person to show the traits of sickle cell disease, they have to get 2 copies of the recessive allele. And so this is a genetic disorder. So this is going to be present from birth. Okay, and so people that have sickle cell often have weakness, fatigue, shortness of breath. Remember that these red blood cells and the hemoglobin that's inside of them are supposed to be carrying oxygen. So these guys aren't getting a very efficient oxygen transport to their tissues. So that's why they're going to feel tired, etc. Plus, your red blood cells are supposed to be these squishy donut-shaped things to fit through these tiny little capillaries. Sickle-shaped cells often get caught in those tiny little capillaries and can be very painful, can cause blood clots, a whole bunch of other problems. Sickle cell is very highly concentrated in some particular areas. So we're looking here in certain parts of like equatorial Africa and the Mediterranean. All right around in here has a much higher incidence of sickle cell disease than in other spots of the world. And that's because in these same spots, there happens to be a high incidence of malaria. So a lot of malaria gives us a lot of sickle cell disease, but the two aren't actually related. So sickle cell is genetic, and malaria is a parasite born from mosquitoes. So we know that mosquitoes don't cause genetic diseases, so what's the possible link here? Well, the key to understanding this is the key to understanding people who are a carrier of the disease. So let's say that this small a is the allele for sickle trait, okay? In order for someone to have sickle cell disease, you have to inherit 2 copies of that recessive allele, okay? So these people aren't doing so well, okay? This person that has 2 dominant alleles, okay, we're not going to include them in our discussion much either, because what I want to talk about are these heterozygous individuals that have 1 dominant allele and 1 recessive allele for sickle cell trait. For some reason that is largely unknown, this heterozygous carrier is immune or shows high resistance to malaria. So this person, even though they don't have sickle cell disease, has an advantage in terms of carrying that sickle cell trait because they're immune to malaria. So these are the individuals that are most likely, again, to survive and reproduce and pass down that trait. Well, what that means is that the carriers are the ones reproducing most often, so of course there's going to be a chance that they have offspring that have sickle cell disease. So that's why we see the continued prevalence of sickle cell traits in countries where there's a lot of malaria, countries where there's no mosquitoes, okay, there's no advantage to having this, and there's only a disadvantage because you might develop or have a child that has that trait, so we don't see that kind of stuff there. Okay, so we keep talking about your base sequences, right? Well, in humans, we have over 3 billion bases or letters, okay? And the entirety of all of those base sequences is called our genome. So all of the A's, T's, G's, and C's. So keep in mind that the genome is just a letter, right? So we know what all of these letters are. We know all 3 billion of them, but we don't necessarily know what all of the sequences do. So how did we figure out what all 3 billion letters are? Well, we first started out using what's called the Sanger method. So the Sanger method is something that you need to know is involved with sequencing DNA, which means to figure out the patterns of A's, T's, G's, and C's. So I could go on for hours about how the Sanger method works, but I'm going to try to keep it real brief here. The first thing I need to do is make many copies. Okay, that's going to involve something called a PCR reaction, which you may already be familiar with. Then I'm going to add special nucleotides that are going to attach to different points along the DNA strand, and they're going to show up under fluorescent lights as different base sequences. So if I read these, okay, then I'm able to tell, well, the first thing in my sequence is an A, and then a G, and then a G, and then a G, well, those are C's. So A, C, 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 G, T, T, A. All right, so I can use that to determine the overall sequence of the DNA. Now, if you think this sounds really time-consuming, you are right. We don't use that method anymore, but that's how we originally started sequencing the DNA. Now, finding out all the sequences in the human genome was the ultimate goal of the Human Genome Project. And the Human Genome Project started in the early 90s, and it's important to note that it was an international effort. So teams of scientists all across the globe came together with one goal in mind, which was to figure out the sequence of A's, T's, G's, and C's in the entirety of the human genome. And in 2003, they accomplished their goal. They found out all the sequences, again, just the letters, not what they did. And what's really cool about this is that they made their findings public. So you can actually go online to the Human Genome Project's database and see all 3 billion letters. And it's been great in terms of making information available to other types of resources that want to use that information to identify genes, genetic diseases, etc. And it showed 2 main findings, okay? They sequenced the genomes of many different people because we're all different, and what they found were 2 things. One was that we're mostly the same. Between 2 humans, 99.9% of those base sequences were absolutely identical. So we're mostly the same, but at the same time, we are all unique, that no 2 individuals outside of identical twins shared the same base sequences. And so since we've been able to figure out those patterns of letters, okay, there's been all kinds of new developments in the world of genetic technology. We've been able to figure out, okay, well, what kind of molecules do healthy people make? Which genes make those molecules? So we can identify the gene where hemoglobin is made from. We can identify the gene where blood type comes from. It's pretty cool. We can find those DNA segments. We can figure out how to copy those genes, and now we can even find a way to distribute healthy copies of those genes into humans that have defective copies and try to help alleviate symptoms of certain genetic or degenerative diseases. But who owns that information? So a couple years ago, there was a really interesting case in the US Supreme Court in which a biotech company was trying to patent a genetic sequence. So somewhere in that 3 billion letter code, they found a specific sequence of letters that coded for a particular gene. And their argument was that, well, since they sifted through all 3 billion letters and they found out what that particular gene was for, then they have the right to make industrial use out of that knowledge, that no one else should be able to use that without paying them a fee. Okay, well, I guess I can kind of see their point, right? Because if these research companies aren't able to make money off of their findings, what do you think the odds are that they're going to go sifting through 3 billion letters worth to find one particular gene? Probably not very good. On the other side of that argument, though, are other biotech companies that are saying, hey, that same gene that you want to patent, well, I can use that in a different way. And if I'm not allowed to use it, then I can't make medicines using that information either. Okay, so their argument was that that patent holder shouldn't even have a patent, that is free and public knowledge. How can you patent someone's DNA? How can you put a patent on someone's identity? So it's a really interesting case there in terms of who owns genetic information. Now, this information isn't just for medical purposes. It's been helping us a lot in taxonomy. And taxonomy is the classification of organisms into different phylums and kingdoms and domains, etc. So back when I was your age, like in the dark ages, there were only 5 kingdoms of living things. So we learned things as either being animals, plants, protists, fungi, or bacteria, because it was assumed that all bacteria were similar. But now that we've been able to sequence genomes, scientists have found that regular bacteria, or eubacteria, are in fact very, very different from another type of prokaryotes called archaebacteria. So they're both prokaryotes, and it used to be that they were all grouped together. Well, through genetic testing, it was found that their genomes are very different. So scientists have now separated them, okay, into 2 kingdoms of their own. Even within a kingdom, we can use genetic testing to find out how closely related two things are in terms of their evolutionary relationships. So cytochrome C is a protein that's found in a whole variety of organisms because it's one of the last proteins that is involved in the electron transport chain for all of my higher level friends that have studied cellular respiration before. So a pretty what we call ubiquitous protein, right? So lots of organisms are going to need that gene, but that gene has undergone several mutations throughout evolutionary history. Well, we can take a look at the base sequences for that gene, and it's assumed that the more bases two organisms have in common, the closer their evolutionary relationship, or the most recent their common ancestor is. Okay, two organisms, like let's say a human and a euglena, a single cell protist, they don't have very much in common. That cytochrome C only shares 56.6% of the bases, which means that they are much more distantly related in terms of evolutionary relationships than a human is with a chimpanzee. So pretty cool stuff that we're doing there with those base sequences. And that about does it for chapter 3.1 on genes.